Okay, so for today, I'll be discussing to you one of the major systems that we will be tackling in developmental biology, which is your central nervous system. Okay, so for the early morphogenesis of the nervous system, of course, we are all aware that for the central nervous system, all of the different um, growing structures are actually coming from the neural tube. Okay, so the neural tube becomes markedly enlarged cephalically, and the expanded portion is the primordium of the brain. So when you say primordium, this is actually, the earliest stage of development of a certain structure. Okay, and then for the caudal part, it is actually the forerunner of the spinal cord. So basically, it will become the spinal cord. Okay, so the caudal part of that neural tube. So from the primer that we had a while ago in our laboratory, we have the three regional divisions of the brain. So the first one would be your prosencephalon, or in common names, we call it the forebrain. It will eventually give rise to its later stages, which includes the telencephalon, which is formed by a mid-portion and two lateral outpocketings, which involves the primitive cerebral hemispheres. And then the second one would be the diencephalon, which is characterized by the outgrowth of the optic vesicles. So these optic vesicles will eventually be, the parts of your eyes. Okay, and the next division of the brain would be the mesencephalon, which is usually called the midbrain. It is separated from the rhombencephalon by a deep furrow called the rhombencephalic isthmus. Okay, so basically the rhombencephalon is now your hindbrain, which give rise to the metencephalon, which later forms the pons and the cerebellum, and then the myencephalon, which later forms the medulla oblongata. So in this figure, you will see the transverse sections through successively older embryos showing the formation of the neural groove, the neural tube, and then the neural crest. Okay, so in here, you will see you are already familiar with the changes, right? So when the neural crest is already being formed, you will see here that the neural cells will eventually detach from that developing um, neural tube. Okay, and then the cells of the neural crest migrate from the edges of the neural folds and develop into spinal and cranial sensory ganglia. So what are these ganglia? So these ganglia are actually groups of neuron cell bodies, and they are usually categorized referring to their primary functions. So for example, we have here the sympathetic ganglion. So more likely it's used, or it will become part of the sympathetic nervous system. And then you also have the pre-aortic ganglion, which will be part of your aorta. Okay, that is included on your cardiovascular system. So depending on what type of ganglia, of course, those ganglias are being referred to by their primary functions. Okay. So in here, you will see the different stages in the formation of the human central nervous system. So A would show you to the age of 22 days. So it would look like this. So I know that you're already familiar with the neural folds, the pericardial bulge, which will eventually become the heart. And then you have your otic placodes. When you say otic placodes, it is part of your ears. Okay, otic. Okay, and then you have the somites, and then of course the cut edge of the amnions. And then in a different view, you will see here the surface ectoderm and then the neural crest. And then letter B, it would show you the difference now when it turns or it ages 23 days. Okay, so you will see the anterior neuropores and then the somites and then the posterior neuropore. Okay, so the numbers here would actually depict also the formation, okay, of a normal fetus. Okay? And then C would be 25 days, and then 28 days for D. So, sorry for this one. This is 25 days, and then this is 28 days. Okay? So, those are the different changes that you will see when it comes to the development of the early stages. Okay? So, may tinatawag kasi tayong early and late stages. So, for example, um, yung katulad kanina sa brain. Okay? So, yung mga tinatawag nating later stages are the telencephalon, myelencephalon. Okay? Pero yung earlier stages nun, kumbaga, those are the primordial stages or the earlier stages. Okay? So in here, you will see the different embryonic vesicles, okay, and then depicting, let us use the colors for us to be able to depict the future structures, okay. So in here, you will see the forebrain or the prosencephalon. The late embryonic stages would be the telencephalon and then the diencephalon. And then the main components would be your cerebrum for the telencephalon. One more. So for the cerebrum, you have the motor area. Okay, so more likely all of the things that you do in a physical manner, okay, like movements, yun yung tinatawag nating motor. Okay, so it controls your voluntary muscle movements. Sensory cortex is also the center of conscious perception of touch, pressure, vibration, pain, temperature, and taste. And it is also where the association areas integrate and process sensory data. Okay? And then for the diencephalon, it would give rise to the thalamus and the hypothalamus. So for the thalamus, it's actually, or it is part of the limbic system. So when you say the limbic system, it includes the structures of the brain that deals with emotions and also memory. Okay? So it integrates sensory information arriving at the thalamus and projects to the cerebral frontal lobes. So next, you have the hypothalamus, which controls the autonomic functions. So when you say naman autonomic functions, this is a control system that acts or takes place without conscious effort. Okay, like for example, appetite drives, like thirst, hunger, and sexual desires. Okay, so when you say the autonomic functions, usually it is known as the fight or flight response. Okay, so it is also responsible for the reproductive behavior. So apart from its autonomic functions, we all know that meron tayong sinasabi na yung feeling or the emotion of love or yung feeling of being loved, hindi siya sa heart. Okay, ina-associate lang natin siya sa heart. But definitely, yung feelings of being loved or yung feeling ng love, usually pinoproduce siya ng hypothalamus. Okay, so there's actually a joke about it na pwede mo daw sabihin sa isang tao na I love you from the bottom of my hypothalamus. Okay? So para din siyang part din ng limbic system. Okay? Together with the thalamus. So more on the emotional side sila of the humans. Okay? So, the hypothalamus also participates in emotional responses. It also secretes ADH and oxytocin. It also secretes releasing hormones for anterior pituitary regulations. Okay? For the midbrain, this one, or the mesencephalon, so the main components in adult forms would be your optic lobes or the tectums and then the midbrain nuclei. Okay, so for the optic lobes, it integrates visual for information with other sensory inputs and relays auditory information. For the midbrain nuclei, it involves involuntary control of muscle tone, processing of incoming sensations, and outgoing motor commands. Okay? So, balikan natin what is the difference of the voluntary and the involuntary. So when you say voluntary, it's something that you can control. So it's with your free will, kaya mo siyang gawin, ikaw ang nagko-control. When you say involuntary, we cannot control them. Okay? Kailangan nilang function sa kanila lang without controlling, like the beating of the heart, and of course, the regurgitation that's happening with your digestive system. Okay? Especially in your stomach. Hindi naman pwedeng sabihin sa stomach na, stomach, wag mo munang gilingin yung mga kinain ko para hindi ako magutom agad. Okay? So, walang control na nangyayari sa mga ganong structure sa katawan natin. Okay? So, next, for the hindbrain or the rhombencephalon, we have first the metencephalon for the late embryonic structure, and it gives rise to the cerebellum, which is involved in the involuntary coordination and control of outgoing movements for equilibrium, muscle tone, and posture. And then we also have the pons, which links the cerebellum with other brain centers with the medulla and spinal cord. It also modifies output of respiratory centers in the medulla. And then next, for the myencephalon, we have the medulla oblongata. It regulates heart rate and force of contraction. And then vasomotor controls. So when you say vasomotor, this is the action upon a blood vessel alter its diameter. So vasomotor, ibig sabihin lumalaki yung diameter ng blood vessels. Okay? Like for example, galing ka sa mainit. Okay? So, ang ginagawa ng blood vessels natin usually nilalakihan yung diameter or nagda-dilate sila para magkaroon ng response yung katawan natin na pababain yung temperature. Okay? So, those are types of vasomotor controls. And then, it also sets the rate of respiration. It relays information to the cerebellum. Okay? So again, these are the early embryonic structures, and then you have the late embryonic structures, and then eventually the adult forms. Okay? So here, it would show you the early morphogenesis of the nervous system. So ito yung sinasabi ko na early development, you have the prosencephalon, mesencephalon, rhombencephalon, and then you have here the cervical flexure that would actually give you the idea na this is actually the part of the head, no? And then, of course, pababa is the spinal cord. Okay, so in an adult form, ito na yung mga derivatives ng inyong mga early morphogenic structures. Okay, so here you already have your telencephalon, the diencephalon, the thalamus, hypothalamus, the brainstem, which includes the midbrain, the pons, and then the medulla. Okay, and then you have the cerebellum or the hindbrain, and then the spinal cord. Okay, so just be familiar with these parts. Okay, so for the histogenesis of the central nervous system, the establishment of the neuroepithelium is very important. Okay, so this is the wall of the neural tube that is made up of the neural ectodermal cells. So as the primitive neuroepithelium matures, it can be subdivided into different layers. So the first layer would be your ventricular or ependymal layer. This is the innermost layer that contains the cells which are still in the mitotic cycle. And then the ependyma, this is actually a columnar epithelium which lines the central canal and the ventricular system of the central nervous system. Okay? And then next, you have the marginal layer. This is the outer cell. This is the outer layer, which is actually cell pore layer, and it includes the white matter. And then the mantle layer, this is the post-mitotic neuroblast, which leave the inner ventricular layer to form an intermediate layer of densely packed cells you call as your gray matter. Okay? So, next we have the neuroblasts. These are initially bipolar in nature. They do have slender processes connecting both central luminal borders and external limiting membrane of the neural tube. They are also the daughter cells that lose their ability to undergo mitosis and migrate toward the outer wall of the neuroepithelium, which is generated by, or which is generating the daughter cells. And they begin to produce processes which are forerunners of axons and the dendrites. Okay? And then next we have the neuroglia, which actually arise from the neuroectoderm. And the glioblast or the glial progenitor cell is the most common intermediate type of cell. So when you say the glioblast, ito yung parang stem cells nila. Okay, yung mga earlier types of, or earlier kumbaga parang parent cell nila. Okay, 'di ba? Ang alam natin sa stem cells, when we have stem cells, they can be transformed into different types of cell depending on which part of the body it is being needed. Okay, or it is being positioned. Okay? And then next we have the ependymal cells. This is thin epithelial membrane lining the ventricular system of the brain and the spinal cord. So this is actually a table of the different cells that you can find in your nervous system. So the cell types, of course, we have different confirmations on how to determine a neuron cell. Di ba may iba-iba silang klaseng configuration. Like for example, you have the multipolar cells. So in this type of multipolar cell, they have many dendrites and one axon, and they function for most of the motor neurons and most of the CNS neurons. And then you have the bipolar, which has one dendrite and one axon. It is found in special sense organs such as the eye and the nose. And then you have the pseudo-unipolar, which appears to have a single axon, which is for sensory neurons. Okay? And then the glial cells, you have the astrocytes, which are highly branched. They actually provide structural support. They regulate neuronal signaling. They contribute to blood-brain barrier and help with the neural tissue repair. And then you have the ependymal cells. They are epithelial-like cells. They line the ventricles of the brain and central canal of the spinal cord. They also circulate cerebrospinal fluid, and some form the choroid plexuses, which produces the CSF. And then you have the microglia, which are small mobile cells. They protect the CNS from infection. They become phagocytic in response to inflammation. And then you have the oligodendrocytes. These are cells with processes that can surround this, several types of axons. And they form the myelin sheaths around axons or enclose unmyelinated axons in the CNS. So as we all know, it's more normal to have myelinated sheets, okay, especially in the axons. It's because for you to be able to have a smooth transmission of responses. Kasi kapag ang inyong axons ay unmyelinated, ang nangyayari dyan, nagkakaroon ng tumatalon yung mga responses. Okay? Or yung mga signals na sinesend coming from the brain. Okay? And then for the glial cells of the PNS or the peripheral nervous system, we have the Schwann cells. So these are single cells surrounding the axons, and they form myelin sheaths around axons or enclose unmyelinated axons in the PNS. Okay, so as you see, or if you observe, there is a difference when it comes to the to which type of glial cells do actually perform the same function. So for CNS, it's the oligodendrocytes that form the myelin sheaths, but for the PNS, it is actually the Schwann cells. Okay, so please take note of this one. Okay, so these are the different or this is the cell differentiation within the neural tube. So this is just an example or a schematic diagram of how it looks like. So this is, if this is the neural tube, of course, you will already see here where you would find the ependymal layers, the mantle layers, the central canal, and then the marginal layers, and then the microglial cells, how they look like. Okay, so from the neuroepithelium, and then or the neuroectoderm cells, it would give rise to different types of cells. Like for example, this apolar neuroblast, it will become a bipolar neuroblast, a unipolar neuroblast, and then eventually it will become a multipolar neuroblast with the presence of several dendrites and one axon. So for the glioblast or the spongioblast, it can differentiate into an astroblast or a oligodendroblast. Okay, so for the astroblast, it can be a protoplasmic astrocyte or a fibrous astrocyte. For the oligodendroblast, it will give rise to an oligodendrocyte. And then the last one would be the ependyma, which can be found also in the epithelium of the choroid plexuses. So, ganito yung itsura ng ependymal cells. Okay? So, next, we have the gray and the white matter in the spinal cord and the brain. So, both the spinal cord and the brain consist of the white matter, which includes bundles of axons, which are coated with a sheath of myelin. And then we have the gray matter, or the masses of the cell bodies and dendrites, which each covered with synapses. Okay, so here you will see the gray matter, okay, and then the white matter, this one. So the white matter has posterior dorsal columns, and then for the gray matter, you have here different horns. You have the posterior or dorsal horns, the lateral horns, and then the anterior horns. Okay? So, hindi ko na masyadong i-discuss sa inyo yung iba't ibang parts of the spinal cord. It's because meron kasi tayong mga tinatawag na tracts. Okay, mga spinocerebellar, spinothalamic, more likely those types of tracts are being used for or are being studied for different reflexes of the body. So, for example, touch, the sense of temperature, okay? Yan usually yung mga parts ng spinal cord natin na tinitignan, lalo na kapag um, nagkakaroon ng trauma. Okay? When you say trauma, for example, may na-aksidente, tapos ang natamaan na part is the spinal cord. So, by knowing which segment of the spinal cord is or did have the trauma, malalaman na ngayon kung ano yung mga possible na effects sa isang patient. Okay? So, yung mga tinatawag nating mga dorsal columns, mga lateral horns, may mga specific sila na mga function na pwedeng i-test to verify if which part of or which section of the spinal cord yung nagkakaroon ng problema. Okay? And then, in the spinal cord, the white matter is at the surface and the gray matter is inside. So, under the microscope, your spinal cord looks like this. So, para siyang may butterfly. So, yung nasa gitna, that is the gray matter. Yung butterfly, parang H na formation, it is the gray matter. And then, outside would be the gray matter. Okay? Or, I'm sorry, it is the white matter. Okay? So, even the brain, it does have the gray and the white matter. Okay? So, kung kanina sa spinal cord, nasa gitna yung gray matter, pagdating sa brain, lumalabas siya, nagiging mas superficial siya compared to the white matter. Okay? So, kanina, di ba, nasa gitna yung gray sa spinal cord. Ngayon naman, yung gray matter, nandun siya sa pinaka-outermost layer of the brain. And then the white matter is inside. Okay, so this is also a different section of the brain which would show you the thalamus, the basal ganglia, and of course the cortex. Okay, so ito, from the level of the spinal cord going up to the brain stem and then to the brain. So ganyan yung itsura niya. May mga tracts tayong mga tinatawag siya. Okay? So, for the development of the brain, the cephalic neural tube by the 4th week of the development has 3 enlargements. So, these enlargements are called again the prosencephalon, the mesencephalon, and the rhombencephalon. During the next week, the prosencephalon enlarges and lateral extensions appear. And these lateral extensions are the telencephalon, and they are connected by a midline structure called the diencephalon. The mesencephalon remains as a single structure, and then the rhombencephalon then develops into into two enlargements, and these are the metencephalon and the myelencephalon. So as these enlargements appear, the brain begins to curve in certain areas. There is a cephalic flexure that appears in the region of the midbrain or the mesencephalon. And then the pontine flexure is located at the junction between the metencephalon and the myelencephalon. It will eventually give rise to the rhombic lip of the metencephalic alar plate and then become the cerebellum. And there is also a cervical flexure that appears at the medulla spinal cord junction. Okay, so other than the cephalic flexure, we also have the cervical flexure. So between 3 to 7 months, all brain structures and nuclei will be differentiated. And then from the telencephalon derives the cerebral cortex. The cortex has been divided into 4 lobes using certain consistently present sulci as landmarks. So yung mga tinatawag natin na sulci, guys, ito. Yan. Yung nasa gitna nila. Yung mga nagdi-divide dun sa mga major lobes na yon. So these lobes are named after the overlying cranial bones. You have first your frontal lobe. Next, you have the parietal lobe, the temporal, and then the occipital lobe. So these are the major 4 lobes of the brain. So the mesencephalon give rise to the midbrain structures, and the metencephalon give rise to the pons and the cerebellum. The myelencephalon derives in the medulla, and the caudal part of the neural tube develops and differentiates into the spinal cord. Okay? So this one is a parang easier diagram for you to understand how the embryonic brain regions develop into the adult forms. So it is alam ko mas effective sa inyo yung parang may nakikita kayong mga kulay to depict yung process of development. So in here you will see the embryo at 1 month. So, yung mga colors na yan, the parang pink one would be the forebrain, violet would be the midbrain, and then the hindbrain would be light blue. Okay? So, at embryo at 5 weeks, magkakaroon ng telencephalon or additional, and then of course the spinal cord. And then this one naman yan. So, from the telencephalon dito na maliit lang, nagiging siya yung parang pinakamalaking part or pinakamalaking derivative pagdating naman sa adult form. Okay? So, pag-aralan nyo lang guys yung flow netong diagram na to, and then for sure if some questions would be given to in your quizzes or maybe in your exams, you would know. Okay? So, pag ang mga tanong sinasabi precursors, ibig sabihin ano yung earlier stage nung mga or pag tinanong it's either precursor or primordia, pag ganun yung tanong, ibig sabihin lang noon, ano yung earlier stage ng isang adult structure. Okay? So, the development of the spinal cord. So, the neural tube consists of 3 cellular layers from the inner to the outer parts. Okay, so first one, you have your ventricular zone. So, the ventricular zone or the ependymal layer gives rise to the neuroblast, which are the future nerve cells, and the glioblast or the future supporting cells, which migrate into the intermediate zone to form 2 collections of cells, which includes your alar plate and the basal plate. Okay, so this is the alar plate, and then this one is the basal plate. Okay, so they are separated by a groove you call the sulcus limitans. So this is the sulcus limitans. Okay, and then next, the intermediate zone or the mantle layer. The ventricular zone cells in the alar plate becomes afferent or sensory neurons and form the dorsal horn of the spinal cord. And then the cells in the basal plate becomes the efferent or the motor neurons and form the ventral horn of the spinal cord. Okay, so ito na yung mga roots. Okay. And then the marginal zone is also known as the marginal layer. Okay. So the two ventral horns bulge ventrally to create a ventral median fissure, and the dorsal horns merge to create the dorsal median septum. So the lumen of the neural tube becomes the central canal of the spinal cord, and the spinal cord extends the entire length of the vertebral canal at week 8 of development. So at birth, the conus medullaris extends to L3 vertebra. In the adult, the conus medullaris now extends to the L1 vertebra. So ibig sabihin, medyo umiikli. Okay, so from L3, pupunta na lang, magiging L1 na lang siya. And then, the spinal lumbar punctures must be performed caudally to the conus medullaris to avoid damaging the spinal cord. Okay. Next is the development of the peripheral nerve. So regardless of their location in the body, most peripheral nerves have similar organizations. So cell bodies of the motor or efferent neurons in the ventral horn of the spinal cord sends out axons that leave the cord through the ventral root and proceed in groups or fascicles towards the periphery of the body, where groups of axons enter and innervate individual muscles. So when you say peripheral nerves, these are the nerves that you see, of course, from the midline going to the sides of your body. Okay? So the cell bodies of the sensory efferent neurons are in the dorsal root ganglion, and this neuron sends processes into the spinal cord through the dorsal root and long peripheral processes or dendrites to the skin or other specialized sensory structures such as the muscle spindles in the skeletal muscles. So near the spinal cord, the dorsal and ventral roots join and the afferent and efferent processes collect into a peripheral nerves. So kung makikita nyo, 'di ba, yung pagkaka-drawing or pagkakagawa ng diagram ng spinal cord, may mga nakikita kayong parang fibers or roots. So yun yung mga tinatawag nating mga dorsal and ventral roots. Okay? So in addition, components of the autonomic nerves pass through some peripheral nerves, and cell bodies of efferent neurons arise in the lateral horn of the gray matter in the spinal cord, and their axons leave the cord along with the motor axons in the ventral root. So they soon branch out, however, into a ramus communicans, which can lead to a purely autonomic nerve or ganglion. So for the axonal growth, so first, the formation of a nerve begins with the outgrowth of axonal processes from some of the large motor neurons within the basal plate of the spinal cord or brainstem. And then the first processes that grow out in the formation of the nerve is called your pioneering fibers, and they typically arise from large neurons. And then the tip of the slender processes of an outgrowing nerve fiber is expanded to form a growth cone from which numerous fine pseudopodial processes or philopodia actively extend and retract. Okay? So in here, yan. Ito yung makikita nyo for day 40 fetus, yan. Yung mga peripheral nerves, ayan, may mga numbers sila. But for, of course, when an organism develops, so the distribution of the nerves are, more likely different. Okay? Yan. So one of these, of course, peripheral nerves that we also know, or types of nerves that we also know are the cranial nerves. Okay? So, the continued production and transport of cytoplasmic materials down the nerve fibers is what you call your axonal transport, and it is a major means by which a neuron is able to maintain continued and adaptive contact with the periphery. So, even in the adult, if a nerve is severed or traumatized, the part of the nerve distal to the cut degenerates, but uh, new growth cones form at the ends of the proximal processes and renewed outgrowth. In this case, called regeneration. So there is also a regeneration part. So for example, from the point where it is being severed. Okay, so it brings the nerve back in contact with its end organ. Okay, so there's always this possibility of regeneration when it comes to this um, nerve growth. Okay, but of course, depending on the severity of the trauma. Okay, kasi 'di ba sometimes if the trauma is too damaging to the nerves, more likely regeneration will no longer happen. Okay, so um, in in medical in medical what call this setting, for example, you will be undergoing surgery. Okay, so usually kapag nagbibigay sila um ng the numbing they call this, pag nagbibigay sila ng mga itinuturok para mag-numb yung katawan, usually ginagawa nila yon above, kailangan above siya nung pinaka nerve, spinal nerve. Okay? Kasi kapag below, no, iba kasi yung conformation or yung paglabas ng roots or nerves ng spine natin. Okay? So for example, sa L1, sabihin natin L1 position or sa yung part ng body mo na sinusuplayan ng L1, kailangan sa taas nun, dun ka mag-i-inject. Okay? Kasi ang labas ng nerves um, ng spine natin, usually lumalabas siya dun sa kasunod niya na level. So, for example, L1 yung pwedeng point of injection mo. Kasi yung kailangan yung body part na magnanumb is below L1. Okay? So, kailangan lagi siyang nasa taas. Kung saang part man ng katawan mo yung kailangan na lagyan ng anesthesia. Okay? Kasi yung roots, again, yung level of, iba yung level nung nerve kesa dun sa level ng paglabas niya from the spinal cord. Okay, laging yung parang paglabas ng mga outgrowths niya, kailangan mas mababa. Okay, so kailangan kung ano yung ini-innervate ng katawan natin sa L1, kailangan yung nasa taas nun yung kailangan mong injekan ng anesthesia. Para sure na magnanumb yung part na yun. Kasi pag nagkamali ka at ikaw ay nag-undergo ng surgery, walang saysay yung anesthesia kung magkakamali yung magtuturok sa inyo. So, kaya nga merong specialization ng anesthesiology when you go to medicine. I think it's around 4 years for you to finish anesthesiology. Okay? Pinag-aaralan yun maigi. Hindi siya basta-bastang specialization. So, development of a nerve to the limb. Okay, so in all, there are about 20,000 motor neurons that send axons into the hind limb. And as axons first grow out from the spinal cord, they soon come together in aggregates and preferentially pass through the anterior portions of the sclerotomes of the somites. So the posterior sclerotome as well as the perinotocordal mesenchyme acts as a barrier for the outgrowing motor nerve axons. So the major nerve trunks leading into the hind limb bud form along the early highways through which the outgrowing nerve processes collect. So even neurites from totally foreign nerves like those that are found in the wings, can follow the main nerve trunk highways as highways in the leg. So sensory nerve fibers also enter developing muscles and late in the prenatal period, but they induce certain groups of immature muscle fibers to form muscle spindles. So other sensory nerve fibers form specialized terminals in the tendons and become Golgi tendon organs, and both this and the spindles acts as stretch receptors or proprioceptors of the muscles. Okay? So in here we have an example. So usually for human beings, the process of the nerve growth for in the limbs is through vasculogenesis. Okay, so when you say vasculogenesis, this is the differentiation of the precursor cells angioblast into endothelial cells during the embryonic differentiation or development. Diba si endothelial cells, they are the, like the stem cells which produce the same, which produces a different type of cell to be able to become functional. Okay, so ito yung mga endothelial cells na to, sila yung parang stem cells on the vasculogenesis to be able to form new cells to form new nerves. Okay? So, dito makikita nyo for this is a progressive vascular remodeling from Carnegie stage 13 through 21. So, the yung mga S na nakikita nyo, it is the subclavian artery. A is for axillary, B is brachial, and then M is median, radial is R, ulnar is U. So, ito nakikita natin sa forearm or sa arm natin. So, I is for interosseous, palmar arches is PA, and then digital is D. So, arteries are colored in red, and then of course, the veins are colored in violet. Okay? So, that's how it looks or how it develops. So, from stage 13, ganito yung itsura niya. Habang humahaba din yung limb, okay, mas dumadami din or mas nagpo-progress din yung process ng vasculogenesis. Okay, and then stage 17 and 19 until mag-stage 21. Okay? So, kung sa humans, meron tayong tinatawag na vasculogenesis. Pag sa ano naman, specific naman na ibang organism like your chick, ang mga chicken, and so they undergo naman what you call the angiogenesis. Okay, so unfortunately we weren't able to to experience yung egg windowing. So usually kasi sa Dev Bio siya ginagawa yung egg windowing, yung magpapagrow tayo ng balot. Okay? So, with angiogenesis naman, this is the formation of new blood cells from pre-existing vessels. So, it can be through sprouting or splitting process. Okay? So, kung sa vasculogenesis, the development of the nerves are actually because of the reproduction of new cells. Dito naman sa angiogenesis, ang nangyayari is from a pre-existing cell, nag-outgrowth yung susunod na nerves. Okay? So, this is an example of the CAM assay for a chick embryo. So, yan. Angiogenic, pagkaganyan. So, from 0 hours, nagiging ganito siya. So, meron tayong mga tinatawag na researches which are anti-angiogenic. Ibig sabihin, you are testing parang the possibility of a certain substance to stop angiogenesis. So, kapag ka kasi, diba, kapag ka yung mga anti-angiogenic studies pinag-aaralan, ito kasi guys yung ina-apply natin kapag ka, for example, ayaw nating mabuo ang isang fetus. So, for example, yung mga iniinom na mga kung ano-ano, yung mga pampalaglag, yun. Parang ganun yung concept ng mga anti-angiogenic studies. Okay? Para hindi mabuo yung mga fetus. Paano mag-stop yung development ng nerves. Okay? Kasi 'di ba sa cheek, yan lang talaga yung way nila para mag-develop. Okay? Para mabuo sila. Okay? Unlike human, sa loob sila ng katawan ng human talaga nagde-develop. And then, regeneration pa ng cells. Ito dito, nagre-rely lang sila sa ability ng cheek na makapag-generate ng mas maraming nerves for blood flow para mabuhay siya kasi nasa eggshell siya, 'di ba? Nagde-determine lang din naman ang growth ng cheek kapag ambient yung environment niya. So, merong kinalaman ng temperature. Okay? May kinalaman din kung ano yung mga kung meron bang application ng mga substances. Okay? Sayang hindi natin nagawa to, but definitely if there will come a time na maganda tong egg windowing. Okay, marami kayong matututunan. So, katulad nito kapag ka-fresh, yan. Kung maglalagay man kayo ng isang substance na anti-angiogenic, so you should expect that hindi magkakaroon ng ganyan na karaming nerves. But if normal yung growth or normal yung process of angiogenesis, yan, tuloy-tuloy yung pagdami nung mga nerves na yan. And then eventually, magkakaroon ng complete embryo by day 28. Okay? So, ganyan yung angiogenesis. But basically, sa angiogenesis, may part or ang initial part pa rin niya is vasculogenesis. Okay, but then yun nga, hindi nagko-continue yung vasculogenesis na yun kasi parang nagtara-transfer siya or lumilipat siya from vasculo to angiogenesis. Okay? So, next for the autonomic nerves. So, when you say autonomic, ito naman yung ito yung sinasabi ko kanina ng mga fight or flight, they are associated with fight or flight responses. Okay? So, accompanying the motor and sensory nerve fibers within the proximal parts of the spinal nerves are efferent and afferent fibers of the involuntary or the autonomic nerves. And then the efferent nerve fibers leave the spinal nerves and make connection with the bodies of the second-order neurons of the neural crest origin, and the cell bodies of the second-order sympathetic neurons are located in chains of ganglia that run on each side of the inner body wall of the thorax and the abdomen. Okay? So, this neuron sends out processes to visceral structures such as blood vessels and skin glands. The afferent fibers return from these structures to cell bodies located in the dorsal root ganglia, and central connections are then made with spinal cord. So, second-order parasympathetic neuron arise from neural crest cells in the cervical and sacral regions, and often migrate considerable distances to innervate structures such as the gut and the heart. So for parasympathetic responses naman guys, ito naman yung tinatawag natin na rest and digest. Okay? So kung ang autonomic ay fight or flight, for parasympathetic responses, it's rest and digest. Usually these are the responses where the body is, relaxed, resting, and in feeding state. So usually it happens after stressful situations. So kung yung fight or flight, yun talagang may stress, okay? Pagka parasympathetic naman, it's more on the resting and feeding stages. Okay? So for the parasympathetic nerves or the rest and digest functions, so ito yung roles nila. So for example, constriction of pupils, stimulate saliva, slow heartbeat, constricting airways, you have the stimulation of activity of the stomach, and then you also have the inhibition of the release of glucose and stimulation of the gallbladder and the stimulation of the intestines. So it also includes the contraction of your bladder and then the promotion of the erection of the genitals. For the sympathetic or the fight or flight responses, you have the dilation of your pupils, So, mas marami siya compared sa parasympathetic. So, you also have the inhibition of your saliva, increase heartbeat, relax airways. Okay? So, please take note of this parasympathetic and sympathetic responses of the body. Okay? So, for the cranial nerves, the spinal nerves are segmentally arranged and all of them are built on the same general plan. So, the cranial nerves, although most likely derived from segmental structures in the phylogenetic past, have lost some of their regular segmental arrangement and have become very highly specialized. So, the cranial nerves can be subdivided into several groups in the basis of their embryological origin and overall function. So the first two cranial nerves, the olfactory and the optic nerve, can be considered to be extensions of the brain tracts rather than true nerves. Okay? And then several of the cranial nerves, 3, 4, 6, and 12, appear to have evolved from primitive ventral motor roots. Nerves 5, 7, 9, and 10 are mixed motor and sensory nerves, and each nerve supplies the derivatives of a different branchial arch. And then the sensory components of the nerves 5, 7, 9, and 10, as well as the auditory nerve 8, have a more complex origin. So neurons of some of the ganglia are derived from neural crest, whereas neurons of other ganglia or parts of the ganglia are derived from ectodermal placodes. So again, with your cranial nerves, there are nerves which are sensory in nature. There are also nerves that are motor in nature, but there are also cranial nerves which are mixed. So they can either be motor and sensory or sensory. Okay? So in here, you will see the different cranial nerves. So for the anterior aspects of the midbrain, you have your olfactory. So this is more likely responsible for the sense of smell. For the optic of cranial nerve number 2, this is for your eyes. And I would just like to give a fact about your optic nerve. This is the only cranial nerve that exits at the back of your brain. Okay? So, kaya majority of the time, if you, or kung napapansin nyo, if you have problems sa mata nyo, usually ang sumasakit sa ulo natin ay yung bandang nasa likod or yung sa may taas ng nape. Okay, it's because your optic nerves exit to those and then umiikot siya paharap. Okay? And then for your oculomotor, ito, cranial nerve 3 and then trochlear nerve for they are usually innervations of your midbrain. For the pons, you have your trigeminal or they are connected to your pons, the trigeminal, abducens, spatial, and vestibulocochlear. And then for the medulla, you have the glossopharyngeal, vagus, accessory, and hypoglossal. Okay? So your assignment for this one would maybe grouping them if they are sensory, motor, or mixed. Okay? So the development of motor function in the human embryo. So many aspects of the development of the neuromuscular system are reflected in the movements of the embryo as a whole. So until about 6 weeks, the embryo merely rocks back and forth passively in its bath of amniotic fluid, with its only intrinsic movements being the regular beating of the heart. Toward the end of the second month, the embryo makes its first spontaneous movements, and these are simple side-to-side twitchings which serve to indicate the functional maturation of the musculature in the body wall. So, actually, about a week earlier, the embryo is capable of making weak twitches in the neck in response to striking the lips or nose with a fine bristle. And this behavioral pattern signifies that the first functional reflex arcs have been laid down. And in succeeding days, the twitching movements in simple reflex capabilities extend progressively caudal in conformance with the general cephalocaudal rule of development. Okay? So, lastly, for the molecular regulation of the CNS development, so these are important. Take note of this, molecular indicators. You have the SHH, which ventralizes the neural tube in the spinal cord region and induces the floor and basal plates. You also have the bone morphogenic proteins 4 and 7, which are expressed in non-neural ectoderm. They maintain and upregulate expressions of the PAX3 and PAX7 in the alar and roof plates and induce the and maintain expression of the dorsalizing genes. You also have the Hox genes that pattern the antero-posterior axis in the hindbrain and specify the rhombomer identity. You also have the LIM1 and OTX2, which are transcription factors containing a homeodomain patterning the antero-posterior axis in the forebrain and midbrain regions. You also have the FGF8, which is secreted by two other organizing centers, the anterior neural ridge and the rhombencephalic isthmus. And then lastly, we have the BF1, which regulates the development of the telencephalon, and the isthmus expresses engrailed genes that regulate differentiation of the cerebellum and the roof of the midbrain. Okay? So that actually ends my presentation for today. If you have any questions, just let me know. Okay? Thank you.